Swayam Prabha. Digital India. Educated India.
Hi everybody, this is Dr. Ravindra Maradi, Associate Professor in the Department of Biochemistry, Kasturba Medical College, Manipal. In the today's class, I will be discussing about sources of ammonia. The topics which I am going to discuss are under the following headings. First, we will see what is ammonia, then when does it produce, how ammonia is produced and where it is produced. Ammonia is produced in different tissues. We will see in different tissues how it is produced. First thing is, what is ammonia? Ammonia is a compound produced during the protein metabolism. As such, when the proteins are digested in the intestine, they are absorbed in the form of amino acids similar to the glucose. It is a secondary active transport. Once the amino acids are entered into the blood, they undergo deamination. Okay? And once that amino groups are taken out from the amino acids, you get free ammonia and keto acids. That is how the ammonia is produced. We will see, and this ammonia is the normal range is very less. The normal level is around 5 to 35 micromoles per liter in the blood. You have to maintain the ammonia level in this narrow range only. If the ammonia level increases in the blood, its condition is called hyperammonemia. This increased ammonia is toxic to the brain. We have to maintain this level so that the body will not suffer from any disorder associated with the increased ammonia level. So, what are the causes for this hyperammonemia? Once the ammonia is produced during the amino acid catabolism, this ammonia has to be detoxified and should be excreted out of the body. So, the major cause for increased ammonia level are, one is this ammonia major part is excreted in the form of urea. So, any defect in the enzymes producing the urea, that is in the urea cycle, that causes increase in blood ammonia level, or any liver pathology, any liver disease, this causes the hyperammonemia. So, what are the effects? Ammonia actually it increases the blood pH and it crosses blood brain barrier and it can cause cerebral edema, tremors, coma and death. That is why we need to take care of this ammonia level. We have to maintain within that normal narrow range. Next, when does it produce? Ammonia, whenever we take a dietary protein, it is digested and then absorbed in the form of amino acids. Once it comes to the liver, the amino group is removed from these amino acids and that is one source of ammonia. And this ammonia can also be produced during the catabolism of proteins. For example, in case of starvation or prolonged fasting condition, body utilizes the proteins, body proteins, they are catabolized into amino acids and the free amino group is removed and the remaining carbon skeleton used as source of ammonia. That is the other source. So, in these two conditions, either we are in well fed condition or fasting condition, we will be getting this ammonia. Now, we will see how ammonia is produced from different amino acids. Almost all the amino acids, the first thing body does is during catabolism, it removes that free amino group which is protecting the amino acid from oxidation. So, that free amino group is released in the form of ammonia, and left out skeleton is called alpha keto acids, that carbon skeleton, and that is used as a fuel to release to get the energy, and also precursors of some of the other compounds like niacin. The free ammonia that is released is highly toxic to the body. It has to be excreted out from the body. And why this ammonia is toxic? Because the ammonia, it increases the body pH and it can cross blood brain barrier to cause cerebral edema, tremors, coma and death. So, the body what it does is it takes that free ammonia and it converts that into urea in the liver, and majority of that, around 86 to 90% is converted into urea and excreted out of the body, and some minor portion is also excreted in the form of ammonium ion in the kidney, which helps in the acid base balancing. You might be thinking that why this if you add up these ammonia that is excreted in urea and ammonium form, it does not come up to 100%. The remaining amount, small amount, is also excreted in some other nitrogen containing compounds like creatine and uric acid. If you see the structure of this urea, it has got two amino group, two amide groups, NH2, NH2 and a carbonyl group. So, this amide group, when this urea is dumped into the blood, it does not change the body pH and it is non-toxic, and it is easily it can cross the plasma membranes, and it does not require any protein to carry in the blood, and it is a very beautiful compound, and that is produced, is excreted out of the body in through the urine. Now, we will see in most of the tissues how this ammonia is produced. In most of the tissues, including skeletal muscles, whenever this amino acids, the first thing body does is removes that amino group in the form of ammonia. Okay. When you remove the amino group, those reactions are called deaminations. Deamination reaction means D means removal of amino group is deamination. Most of the tissues, the free amide group is removed in the form of ammonia, and that is taken up by the glutamate to convert into glutamine. The glutamate can take up one amino group, and alpha ketoglutarate also can take up one amino group and ammonia and convert into glutamate, and glutamate converts into glutamine by the glutamine synthetase. It is the ATP requiring reaction, and this glutamine is dumped into the blood, and this is taken up by either intestine or kidney, and then further catabolism takes place. So, glutamine is a very important compound because it does not change the body pH, because that ammonia is present in the amide group, the nitrogen is present in amide form, that is why it does not change the body pH. So, in kidney what happens, the glutamine that is present will be, that is coming from most of the tissues. As I told, that ammonia is carried in the form of glutamine, because the ammonia, once it is dumped into the blood, it increases the body pH. That is why the transport form is glutamine. So, glutamine, it is having an amide groups and it does not change the body pH. That is why this is the transport form. Once in the kidney, it is seen by an enzyme glutaminase, and this glutaminase enzyme removes that ammonia and forms glutamate and ammonia. This ammonia combines with a proton to form ammonium ion and excreted in the urine, and it plays a very important role in acid base balancing as it helps in excreting a proton. In case of intestine, what happens? The glutamate that is coming from most of the tissues or from the dietary protein, two sources. The glutamine again is seen by glutaminase enzyme and takes out that amide group of glutamine and converts into ammonia and glutamate, and this ammonia is then taken up by the liver through the portal blood, and this ammonia is also produced by the action of intestinal bacteria. In the bacteria, the ammonia is produced is also a source, and that also along with the ammonia of glutamine it goes to the liver. This is the other source. And the urea that is excreted, that is present in the blood, a part of urea can be taken up by the intestine, and it sees an enzyme urease and converts that urea into carbon dioxide and ammonia. So, this is the other source of ammonia from urea. And what happens in the muscle? There are a group of enzymes we call that as aminotransferases. These aminotransferases, they transfer, as the name suggests, they transfer a amino group from the amino acids, and that is converted into amino acid is converted into alpha keto acid. That amino group from amino acid is taken up by the alpha ketoglutarate and forms glutamate, and this reaction is catalyzed by PLP, that is pyridoxal phosphate, vitamin B6. This glutamate again sees one more aminotransferase, that is alanine aminotransferase. This is also PLP dependent reaction, and the amino group from glutamate is given to the pyruvate, and pyruvate is converted into alanine, and glutamate is converted into alpha ketoglutarate, and this alanine goes to the liver. Again, the amino group from amino acids in the muscle is not directly dumped into the blood, that is coming through alanine. The reason is here alanine again it does not change the body pH. That is why body knows its biochemistry, it sends the amino groups ammonia in the form of glutamine or from the alanine that goes to the liver. So, once they come to the liver, here we have the alanine, and that is again acted upon by the alanine transferase. Here it is a PLP dependent enzyme. So, alanine gives that amino group and it is converted into pyruvate. The amino group is taken by alpha ketoglutarate and converted into glutamate, and here the glutamate is seen by glutamate dehydrogenase enzyme, and that gives the ammonia, free ammonia, and that enters into the urea cycle. That is the first source of ammonia. Then that glutamate can also be acted upon by one more transaminase, that is the aspartate transaminase, and here this glutamate gives that amino group to alpha oxaloacetic acid OAA to convert into aspartate, and glutamate converts into alpha ketoglutarate, and aspartate is the source of second nitrogen of the urea. So, then when you see that urea structure, it has got two amino groups. The chemical structure is NH2C double bond O NH2. So, two nitrogens are donated by one from this free ammonia from glutamate, and another the nitrogen is from aspartate, and CO comes from carbon dioxide. So, in the liver, that is urea is synthesized and excreted into the urine. So, the major sources of ammonia from different tissues we saw, but there are also some minor ammonias. They are produced small amount of ammonia from purine and pyrimidine catabolism. In purine catabolism, we can get this free ammonia from adenosine monophosphate is acted upon by adenosine monophosphate deaminase, and it is converted into inosine monophosphate. During this process, the ammonia is released, and then we also get free ammonia from adenosine acted upon by adenosine deaminase enzyme, and it is converted into inosine. As the name indicates, deamination, the removal of a amino group from adenosine to inosine, and also guanine acted upon by guanase, which releases ammonia, and guanine converts into xanthine. And also pyrimidine ring, when it breaks down the pyrimidine ring, it gives out the ammonia, and pyrimidine is converted into beta alanine and beta amino isobutyrate with the release of the ammonia. And also small amount they are produced from the amines from the diet, and also monoamines like epinephrine, norepinephrine, dopamine, they undergo catabolism by the monoamine oxidase enzymes, giving out the ammonia. So, we discussed about the different sources of ammonia. We started with what is ammonia. Ammonia is a byproduct that is produced during the protein catabolism and amino acid catabolism. And when does it produce? It produces ammonia during well fed or in the fasting condition also. Whenever the dietary proteins they undergo digestion, amino acids absorbed, and when they come to the liver, the first thing body does is removes that amino group in the form of ammonia. And how it is ammonia produced? By different tissues, we saw that it produces by the deamination and then transamination reactions, and in the liver it is the glutamate dehydrogenase enzyme, different enzymes in different tissues, even in the intestine, kidney by glutaminase enzymes. In different sources of this ammonia is produced by different substrates. And then where does it produce? I told in different tissues, and this ammonia is very toxic, and it should be excreted in from the body, and major chunk of this is excreted in the form of urea, around 86 to 90%, and excreted in the urine, and around 3% as ammonium ion. So, that is the summarizes what are the different sources of ammonia. I hope this lecture helped you to understand the different sources of ammonia. Thank you.